Hi, my name is Grace Poon. I'm a scientist on the Cell Separation Research and Development team at STEMCELL Technologies. At STEMCELL, we actively explore ways to efficiently isolate cells from various tissue sources to facilitate your research. Today, I'll be walking you through a scientific poster on the isolation of tumor-infiltrating leukocytes, often abbreviated as TILs, from mouse tumors using our EasySep technology. Here, TILs are targeted by the common leukocyte marker CD45. This poster is based on an abstract submitted for the 2020 AAI meeting, and this method has been further developed into a kit that will be available for everyone to purchase. Let me start by sharing our rationales for working on this project. TILs consist of highly diverse leukocyte subsets with major roles in cancer immune surveillance. It can be challenging to isolate or analyze TILs with sensitivity and precision due to their relatively low frequency, tumor heterogeneity, and the abundance of tissue debris in tumor samples. To address this challenge, we have developed a simple method for isolating CD45 positive TILs from mouse tumors by immunomagnetic separation. There are many advantages to working with an enriched sample. For instance, in the case of single-cell sequencing or flow sorting, TIL enrichment means fewer cells to process and potentially better resolution. Having an enriched TIL population will likely improve the quality and efficiency of your downstream application. Three commonly used mouse models, namely the B16 melanoma, CT26 colon carcinoma, and 4T1 mammary tumor models were selected for protocol optimization. Solid tumors were induced by subcutaneous implantation of B16, CT26, and 4T1 cancer cell lines into syngeneic recipients. To start, you will need to generate a single-cell suspension from solid tumors. Briefly, prepare tumor digestion medium consisting of collagenase, hyaluronidase, and DNase. Mince the tumor tissue into small pieces using a razor blade, scalpel, or scissors, and transfer it into a 14 ml round-bottom tube containing the tumor digestion medium. Incubate at 37 degrees for 25 minutes on a shaking platform. Then, filter the digested tumor sample through a 70-micron cell strainer to obtain single-cell suspension. Spin down the cells and carefully remove and discard the supernatant. Add ammonium chloride for 5 minutes at room temperature to remove red blood cells. Top up with EasySep buffer and centrifuge at 300 G for 10 minutes. Carefully remove the supernatant and resuspend cells at the desired concentration in EasySep buffer, and the sample is now ready for TIL isolation. The TIL isolation protocol we will now review is compatible with most tissue dissociation protocols, so you can easily apply it to your current workflow. The first step of TIL isolation requires the preparation of the TIL isolation cocktail, which consists of 3 components: component A, B, and C. Mix them at a 1:1:6 ratio in sequence and add it to the sample. After 5 minutes of cocktail incubation, add dextran rapid spheres. This allows the TILs from tumor samples to be labeled with an antibody complex that links CD45 positive cells to magnetic particles. Then separate the TILs using an EasySep purple magnet, applying 4 rounds of 5-minute magnetic separation. TILs will remain in the tube while unwanted cells are being poured off. You can resuspend cells in the medium of interest, and your sample is ready for downstream applications. Now I will walk you through some of our results using this method. In Figure 2, we show a comparison of the CD45 TIL frequency before and after EasySep isolation in 3 tumor models. We have selected different start cell concentrations for each tumor model, and this is to achieve higher TIL purity and also taking into consideration the average sample size for each model. Results from B16 samples are shown in the first row. In the start, there are various populations visible in the forward and side scatter plots, and it can be hard to identify the right cell gate for TILs. TIL isolation makes the leukocyte population more visible and often reduces the amount of debris in the sample, which tends to be smaller in size. To assess TIL purity, we first apply a cell gate, followed by a viability gate, which gates on Pi-negative populations that is not shown here. CD45 frequency is then analyzed within the viable gate. Using a single cell suspension from a B16 sample at 100 million cells per mil, CD45 positive TIL frequency in the start and final isolated samples are 14.8% and 95.2% respectively. For T1 samples were adjusted to 40 million cells per mil, and TIL purity improved from 37.6% to 93%. In the third row, we have data from CT26 tumor, starting at 25 million cells per mil. The purities of the start and final isolated fractions are 32.3% and 84.6% respectively. Figure 3 summarizes the performance across multiple experiments. Overall, we observed consistent enrichment of TILs across all 3 models tested, achieving up to 98% in purity. Tumor size can vary significantly depending on the type of tumor model, experimental treatment, as well as the experimental endpoint. To accommodate a range of sample sizes, we optimized the protocol in different magnet types that can hold 5 ml or 14 ml tubes. EZ8's magnet that enables easy isolation of multiple samples at the same time was also tested here. In Figure 4, you can see comparable enrichment of TILs from B16 samples across different magnetic platforms, including the purple, silver, and EZ8's magnets. Not all applications require high TIL purity. Sometimes TIL enrichment with a better recovery is more useful. We found that by simply adjusting the start concentration and volume in a given sample can yield higher purity or recovery. First, let us look at the impact of cell concentration. In B16 samples, starting with a higher cell concentration at 50 to 100 million cells per mil results in higher purity. Figure 5a shows a correlation between high start cell concentration and high TIL purity. If higher recovery is desired, resuspend cells at a lower concentration, in this case 10 to 25 million cells per mil. In Figure 5b, you can see a clear trend where recovery increases as the starting cell concentration decreases, plateauing at 25 million cells per mil. Adjusting the cell concentration means it will lead to changes in start volume. To determine if start volume impacts performance, we kept the cell concentration constant at 10 million cells per mil and modified the sample volumes. These results are shown in Figure 5c and 5d. While there seems to be a trend for a slight drop in purity and increase in recovery as start volume increases, both purity and recovery remains relatively stable across 0.1 to 2 ml start volumes. Overall, this is a flexible protocol that can be used to obtain a higher TIL purity or recovery by simply adjusting the start cell concentration. Furthermore, based on the range of cell concentrations and volumes tested, this protocol can accommodate a wide range of sample sizes. To ensure that we recover all immune cell subsets, we compared the composition of TILs before and after EZ-sept-TIL positive selection by flow cytometry. We first analyzed the B and T cell frequency using anti-CD19 and CD3 antibodies. Then we gated on CD3 positive, CD19 negative T cells to further analyze the distribution of CD4 and CD8 T cells. Last but not least, we analyzed GR1 positive, CD11b positive within the CD3 negative, CD19 negative population, which would include neutrophils, monocytes, and myeloid-derived suppressor cells. Overall, all of the subsets in the start sample were present in the isolated sample. There are differences in frequency, which may be attributed to a greater number of TILs collected in the isolated condition compared to the start. This brings me to the end of this virtual poster presentation. In conclusion, EasySep Mouse TIL CD45 Positive Selection Kit enables the isolation of leukocytes from solid tumors in as little as 30 minutes, while achieving purity as high as 98%. The isolation protocol can accommodate different sample volumes and cell densities that results from tumor tissue heterogeneity. TIL purity or recovery can be optimized by adjusting the start cell concentration or volume. Importantly, TIL immune cell populations of the starting sample are maintained following EasySep isolation. Thank you for listening, and I hope you found this presentation useful. If you would like more information about this product or the data presented here, please visit STEMCELL.com or contact your local sales representative.